Hello, everyone. Can you hear me? Hello? Can you hear me? Hello? What's this? Can you hear me? Is my voice heard? I can't hear you. Hello? Farouk, can you hear me?
Yes, Miss.
Okay, now I want you to prepare paper 1. We will try to solve as much as we can questions on the lesson enzymes. Okay? Let's start. This is which page? Let me see which page is that. Okay, 1 minute.
Doctor, it's 188.
Okay, very good, 188. Fine, let's now paper 1. This is not a very nice paper, so I highly advise you that you answer on a separate paper because I want you to solve it 2 times. Solving it once is not enough. The good thing about paper 1 is that a lot of the questions are repetitive. You will not find repetitive questions in the classified because I try as much as I can to remove the repetitive questions, but you will find them in different versions of the same exam. Okay, so paper 1 needs to be solved 2 times, I'm telling you. Now let's start. Okay, let me activate my pen. This is June 2017, page 188, okay? Bacterial cells divide by a process called binary fission. Which macromolecules must be synthesized for binary fission? Unfortunately, you will not be able to answer this question because this is biological molecules, the lesson biological molecules. But macromolecules are molecules that are large in size and they have a high molecular weight. And the examples that we will study for macromolecules in the next lesson will be the RNA and the DNA. So the answer has to be including 1 and 2. The answer has to be B. Okay? I will not blame you for this question because this is biological molecules. Let's go to question 14. Question 14. A student carried out experiments to investigate the effect of enzyme concentration on the rate of hydrolysis or breakdown of protein in milk. When the enzyme and the milk were mixed, the protein was hydrolyzed. Hydrolyzed means the protein was broken down using water, and the mixture changed from cloudy to clear. The student investigated 5 different enzyme concentrations and recorded the time taken to reach the end point for each. What is an appropriate control for this investigation? Okay, read A, B, C, D, and Farouk, give me an answer. Now, this has got nothing to do with the lesson, by the way. This is paper 6. When I have enzymes, what did you say, A? Yes,
Yeah.
are correct. I hope that you did not look at the mark scheme because I was just going to tell you that when you have any experiment that involves enzymes, the control will be either boiling the enzyme or replacing the enzyme with water. So the answer is yes, A. Okay, very good. Now next. What determines the specificity of an enzyme? In other words, what makes it specific? Look at A, B, C, and D. Look at 1, 2, 3, 4, and 4 first. The bonding between the R groups of the polypeptide. Oh, unfortunately, this is also biological molecules. Let me explain it because we will take it in the next lesson. The molecular structure of a single amino acid, this is something that you will have to learn, is a central carbon atom bonded to 2 functional groups. One functional group is known as the carboxyl group, COOH, and another functional group known as the amine group, NH3, and then a hydrogen from one side, and then a variable R group. This R group is known as the variable R group because this is the group that is different from one amino acid to the next, okay? All amino acids have this part constant. What is the variable part? The R part. And the bonds between the R groups make the enzymes specific. The bonds between the R groups give the three-dimensional shape of the enzyme. We will study this in the next lesson when we study proteins. So I do not expect you to know the answer of number 15. Number 15, the answer is only 1. Okay, the optimum pH of the enzyme does not give it the specificity. Specificity means the enzyme is specific in shape and it gives or it binds to a specific substrate. What makes this enzyme with a specific three-dimensional shape? The bonds between the R groups. Okay, so the answer should include only number 1, which is C. So again, I do not expect you to know the answer to this question. But 16, I think you should. Let's read 16. The graph shows the effect of temperature on the rate at which the enzyme in a biological washing powder digests and removes fruit juice stains. So I have the rate of the reaction on the y-axis and the temperature on the axis, on the x-axis, and a typical graph. Which statements explain the shape of the graph at temperature higher than x? This is a very easy question. A, B, C, or D? Read it. I'll give you a minute. Who do I have? Let me see. Kenzie. Tell me, Kenzie. At this part of the graph, the temperature is very high. Okay, let's see 1, 2, 3, 4, and which are correct. 1. The bonds are broken between the R groups of the amino acids and the polypeptide chains of the enzyme. At high temperature, are the bonds broken? Yes or no?
Yes. Yes.
Yes, of course. So 1 has to be in the answer. Okay, you know, if I am you, if I am sure that 1 is correct, then 1 has to be in the answer. So the answer is either A or B or C. The answer is not D, because there is no 1 in D. So let's look at number 2. There are more collisions between the enzyme and its substrate.
No, because the enzyme is denatured.
Yes, there are, like the enzyme is denatured. It's not going to collide. Okay, so 2 is most probably not in the answer. So this will help me also remove A and B.
And B.
What do I have left? C. C says that 1, 3, and 4. Let's make sure that 3 and 4 are correct. The tertiary structure of the enzyme is altered. We still did not study the tertiary structure, but the structure of the enzyme in general, will it be changed at high temperatures?
Doctor, can you repeat the question again?
The question is right there, here. You mean 3 or the question itself? We are looking at the enzyme at very high temperature at this part of the graph. We want to see what comments or statements are correct. Number 3.
1, 3, and 4.
Yes, but we want to study and understand why. At 3, the shape of the enzyme changes? Yes or no? At high temperature?
Yes.
Yes, because the bonds break at 4. Or let's look at number 4. The shape of the active site and the substrate are no longer complementary?
Yes.
Yes, it's very easy. This question is very easy. So the answer is C. Okay. Okay, did we get them correctly? B, A, C, C? Go back.
Yes.
Okay. Fine. Now the next exam. The next exam, June 2017. Let's look at June 2017. Okay. The enzyme invertase catalyzes the breakdown of sucrose to glucose and fructose. 3 different enzyme inhibitors of invertase X, Y, and Z, so X, Y, and Z, were investigated. The percentage inhibition of invertase was measured at different concentrations of inhibitor. The graph shows the result of the investigation. You have to look closely at the graph. What do you have at the x-axis? The concentration of the inhibitor. This is not the graph that we studied in class. On the x-axis, I don't have the substrate concentration. No, I have the concentration of the inhibitor. And how this affects the percentage inhibition. So put this in mind, okay? Now let's read which are valid conclusions from these results. 1. The higher the concentration of inhibitor X, the less sucrose is broken down. Now look at inhibitor X. when inhibitor X, when its concentration is increasing, what is happening to the percentage inhibition? They are, someone is speaking, but their voice is very low. Increase the volume, please. Inhibitor X, from this graph, we know that inhibitor X is the most inhibitor having the most effect, the most percentage inhibition. Inhibitor Y is less stronger than inhibitor X. Inhibitor Z nearly did not affect the reaction because inhibitor Z, as you increase it, the percentage inhibition will remain constant. But in inhibitor Z, this is very strong. The more it increases in concentration, the more it will inhibit the reaction. So number one, the higher the concentration of inhibitor X, the less sucrose is broken down. Is one right or wrong? The more we add inhibition, the less sucrose is broken down. Right or wrong?
Yes, it's right.
Yes, it's very right. Okay, so one has to be in the answer. This will make me put in mind that either
A and B.
A or B. So I will exclude C and I will exclude B. Okay, this helps me to narrow down. 2, the production of glucose and fructose using inhibitor Y, this one, is higher than when inhibitor Z is used. The production of glucose and fructose using inhibitor Y is higher. Is it higher or lower?
It's lower.
Lower, because inhibitor Y is very strong. It is stronger than Z. So actually inhibitor Y will lower the reaction. So 2 is wrong. This means I will exclude what?
2.
Yes, I will exclude 2. Then the only answer I have will be B. These questions, I advise you to answer them this way. Narrow down until you have one final answer. Let's read 3 because 3 must be wrong. The production of glucose and fructose at an inhibitor concentration of 2 arbitrary units is lower than at an inhibitor concentration of 4 arbitrary units for all inhibitors. Now let's look at this. The production of glucose and fructose at a concentration of 2 is lower than at an inhibitor concentration of 4 for all inhibitory units. Now this is not logic because this means that for all of them at 2, is it all, are they, you know what this means? The production of glucose and fructose. This means that the reaction rate at an inhibitor concentration of 2 is lower than an inhibitor concentration of 4 for all inhibitors. This is not true because at inhibitor concentration of 2, the percentage inhibition was very high at Y and it was lower at Z and it was very low at Y. At 4, inhibitor here, Y and Z are the same. Can you see the meeting point here? Can you see the meeting point between Y and Z at inhibitor concentration of 4? So it is not lower. At inhibitor concentration with a value of 4, inhibition is the same using inhibitor Y and inhibitor Z. Do you see that? Do you see the meeting point here? Are you all with me?
Yes, doctor.
Okay, so 3 is right or wrong?
3 is wrong.
3 is wrong. So the only answer that is correct is 1, which is B. Okay, we will check the mark scheme. Hopefully we will be correct. Now 11. The following statements are about enzymes. Which statements are correct for the induced fit hypothesis? So we want correct statements for the induced fit. Let's see. Folding of an enzyme molecule causes the formation of the active site. Let's see number 2. The shape, by the way, number 1 is right. Okay, we will study the protein structure. The proteins, when they fold and they form a precise 3-dimensional shape, this will give the final shape of the protein or the enzyme and give the final shape of the active site. Let's look at 2, 3, and 4. The shape of the active site changes to enable the substrate to bind. Is this correct?
Yes, it's correct.
Yes, it's correct. 3. Temporary bonds hold the substrate in the active site. Correct?
Yes.
Of course. 4, more enzyme substrate complexes are formed at the optimum temperature. Is this correct?
Yes.
Yes. Okay, I don't have any answer here that says 1, 2, 3, and 4. You know why? Because they want only the statements that refer to the induced fit hypothesis. Only this hypothesis. So which one of those, or how many of those are only related to the induced fit?
C.
Yes, you are correct. 2 and 3 and 4. 1 is a correct scientific sentence, but it's got nothing to do with the induced fit. When enzymes fold, they form the active site. Okay, but the enzyme here is not bonding with any substrate. It's just making its shape. So C is the correct answer because it is only related to the induced fit. I hope you all understand this question. 12. How is the Michaelis-Menten constant used? Now, if I am you, I'm going to look for one word. Why do we study the Michaelis constant? Excuse me? Someone is speaking, but their voice is very low.
Affinity.
Yes, so I will look for this word, and without thinking, it's going to be B. So let's look at the answers. Are we correct? B, C, B? Did we
Yes.
correct any mistakes? Okay, very good. The graph shows energy changes, and I don't want you to write on the classified because I want you to solve 2 times. Now, the graph shows energy changes in a chemical reaction. Of course, this graph, you should know this is for which energy? The activation energy. We did not study the lesson yet. What is the activation energy when the enzyme is added? A or B or C or D? The enzyme, what is the activation energy when the enzyme is added? What is the activation energy when there's no enzyme? The enzyme will lower or increase the activation energy. Wake up with me.
Enzyme lowers the activation energy.
Very good. So without the enzyme, this is the graph. The activation energy will be this distance, 1 plus 2. When we have the enzyme added, what will be the graph? The one below it. So what will be the activation energy?
2 only.
2. Very easy. Okay.
In progress.
The graph shows the results of investigations into the effect of amylase on the hydrolysis of starch at 3 different temperatures. So looking at the graph again, always look at the X at the Y axis. You have time on the X axis and the concentration of the sugar on the Y axis, okay? Concentration of the sugar. The sugar is the substrate. Put this in mind, not the reaction rate. In each investigation, the concentration and volume of the solutions were kept constant. Which conclusion may be drawn? So the independent variable here is the temperature. Which conclusions can be drawn? Let's see. 50 degrees is the optimum temperature. Is 50 degrees the optimum temperature?
No, it's not.
Why do you say it's not?
Because it's the enzyme is denatured.
The enzyme's denatured. The enzyme actually, with time, the concentration of the reducing sugar increases until the concentration of the reducing sugar became constant. The optimum temperature here, they only tried 3 temperatures. What if the optimum temperature is 40? What if they did an experiment at 40 degrees and the graph looked like this? And there was more concentration of reducing sugar. So you cannot be sure that A is correct. A might be correct and it might be wrong. So A is not definitely correct. Okay, so I would put next to A a question mark, but most probably it's wrong. B, at 60 degrees, all the starch is hydrolyzed after 30 minutes. Okay, at 30 degrees, the concentration of sugar became constant. But can you confirm that all the sugar has been broken down? What if the enzyme was denatured before the sugar was broken down? So is B definitely correct?
No.
No. Also, I'm not sure. Now let's look at C. At 70 degrees, the amylase is denatured before hydrolysis is complete. Now let's look at 70 degrees. 70 degrees is the highest temperature. And in a very short period of time, the concentration of the sugar increased and then it remained constant. So what does this show you? That the sugar is being broken down or not breaking down anymore?
Not breaking down.
Okay, so C is most probably correct or incorrect?
Correct.
Most probably C is correct. Let's look at D. D has to be wrong if we are to confirm that C is the answer. The rate of hydrolysis of starch is faster at 50 degrees Celsius than at 70 degrees Celsius. The rate. How do we figure out the rate? We have to draw a tangent passing through the curve and calculate its gradient, okay? So the rate of the hydrolysis of starch is faster at 50 degrees. The tangent would be something like this, than at 70 degrees. Without even measuring the gradient, which graph is steeper? The graph 50 degrees or 70 degrees? 50 degrees is down there, while the 70-degree graph, it's up there. So which graph is steeper, the 70-degree or the 50-degree?
50 degree.
Steeper, Maryam. Steeper means
70 degree.
the 70. So this is steeper. This means that the initial rate of the reaction was very fast. Quickly, the sugar was broken down, but also very quickly the enzyme denatured. It did not complete. You understand? So D is also wrong. So A, we cannot be sure that 50 is the optimum temperature. A is wrong. B, at 60 degrees, all the starch is hydrolyzed. How do you know that all the starch is hydrolyzed? What if some of it was hydrolyzed and then the enzyme got denatured? So also B is not confirmed. C is the most correct because at 70 degrees, the enzyme is denatured before hydrolysis is complete. This is the most logic answer. To me, at least. Anyone has any objections? Hello? Anyone wants to say something? Abdullah, do you have any questions? Sireen, you have any questions?
No, thank you.
Okay, let's move on. The graph compares the effect of temperature on the activity of the enzyme, papain, when free in solution and when immobilized in alginate beads. So this is the activity of the enzyme here, and this is the temperature. At first, obviously, the activity of the enzyme is constant, and then the activity will start to decrease when it is free, and it will take a little bit more temperature for the enzyme's activity to decrease when it is immobilized. Which statement about the effect of immobilization is correct? Basma, can you give me an answer? Immobilization, what does it do to the enzyme? It changes the shape of the active site at higher temperatures, or it decreases the activity of the enzyme at higher temperatures, or it increases the stability at higher temperatures, or it reduces the number of collisions with the substrate. What did we take about immobilization? One of the advantages. Hello? Who will give me an answer? Abdullah, you were not here last class. Maryam, tell me, you were not here last class. We studied immobilized enzymes and free enzymes. We said immobilized enzymes are the enzymes that are attached to a substance and they're not free to move.
Okay.
Okay, and this gives the enzyme advantages. Maryam, what is one of the advantages found here right in front of you? A, B, C, or D?
C.
Yes, it makes the enzyme more stable, more thermostable. Okay, so the answer 12 is C. So it should be B, C, C. Are we correct? Did we do any mistakes?
No.
No.
Okay, good. We can do mistakes, عادي, يعني everyone does mistakes. Lysozyme is an enzyme found in tears and in saliva. Then, which statement refers only to the tertiary structure of the lysozyme? I do not expect you also to know the answer to this question because this is the next lesson. We will study the structure of proteins. It depends on the sequence of amino acids. So a section of the molecules consisting of, no, it's not a single, it will be the sequence of amino acids. Most probably it's D. Okay, we'll take it next lesson. Some RNA molecules called ribozymes can catalyze enzymes in a similar way to protein enzymes. Most of these ribozymes have other RNA molecules as their substrates and catalyze reactions that break specific sugar phosphate bonds in the substrate molecules. We still did not study RNA, but let's try to solve this question. Which statements about these ribozymes are correct? They're telling you that these structures called ribozymes, they are RNA molecules and they work in the same way as enzymes. So read with me. Which statements about these ribozymes are correct? Hydrogen ionic and disulfide bonds will be involved in the ribozyme structure. Okay, we still did not study biological molecules also. We will study that proteins are held together by these bonds, hydrogen, ionic, and disulfide bonds. So these bonds are found only in proteins, not in RNA. So one is definitely wrong. When one is definitely wrong and you are sure, you will exclude all the answers that has number 1. So you will exclude A and B and C. The only correct answer must be D. Let's read number 2. The active site of this ribozyme is formed from a specific sequence of nucleotides. Doesn't this sound correct? Didn't we say that the 3-dimensional shape of the protein depends on the sequence of amino acids? I think we took this from last year. Didn't we say this? Year 11, are you confused or you're with me? Hello?
Hello.
Hi.
Yes.
Anyone has any problems? From last year, and we will take it again in the lesson biological molecules, we know that the enzyme has a 3-dimensional shape. Yes, don't we all know this? Are you all with me? I want to hear your voices.
Yes.
Yes.
Okay, what is holding the precise 3-dimensional shape in the enzyme? These enzymes are proteins. They are made up of smaller what?
Amino acids.
Very good. Amino acids that are bonded together in a particular sequence that will give them a particular shape. This question is telling us that there is a molecule known as the ribozyme. This ribozyme, it is not made up of protein. It is made up of RNA nucleotides, and it is similar. So what is the similarity between this molecule and the protein? 1, these bonds are present. No, these bonds are not present in the RNA molecule because these bonds are present only in the protein molecules. I don't expect you to know this now. But let's look at number 2. The active site of this molecule is formed from a specific sequence of nucleotides. Yes? Why yes? Because the 3-dimensional shape of the enzyme is formed from a specific sequence of amino acids. So therefore, the 3-dimensional shape of this RNA molecule is made up from the specific sequence of its monomers, which are the nucleotides. Do you understand? Who is confused?
Yes.
Anna, can you please explain it again?
Okay, Hadr, I'll explain it again. Okay, I don't actually expect you to know it because this is, again, biological molecules, but I want you to understand. Let me just erase this. Okay, look, my dear. Very, very soon, we will study the structure of proteins. Proteins, they have a specific structure. And why do they have a specific structure? Because they are made up of smaller amino acids bonded together in a specific pattern. When they are bonded together in a specific pattern, they will generate the 3-dimensional shape of the protein. Okay? The molecule that they're talking about is an RNA molecule. The RNA molecule is a large molecule that is also made up of smaller subunits. These subunits are called RNA nucleotides. Remember the nucleotides in DNA? This RNA, they're telling you it also has a precise 3-dimensional structure. Any molecule that has a precise three-dimensional structure, this means that it is made up of smaller molecules arranged in a certain manner. Again, these smaller RNA nucleotides are arranged and bonded in a certain way to create a precise three-dimensional RNA molecule. So that is why it is similar to the enzyme that we are talking about. Clear? Better?
Yes.
Okay. I want to find a way to start a new screen without having to erase one by one. Okay, going back to my, I'll discover this later. Okay, so the answer has to be, what am I talking about? You confuse me. Yes, 2 and 3. Let's look at 3. Ribozymes can form because RNA have a specific shape. Forget the term secondary and tertiary. Doesn't the enzyme have a specific shape also? So 3 is correct or wrong?
It's correct.
It's correct because it's like the, what is called the enzyme. Enzyme have a specific shape and RNA have to have a specific shape. So D. This question is easy, by the way. You think it's hard because you did not take it. The moment you read number 1, you will know that number 1 is wrong because these bonds, we will study in another lesson that these bonds are found only in proteins. So you will say 1 is wrong. The moment you are confirmed or you're sure that 1 is wrong, you will exclude A and B and C, and you will only have D actually. So this question is not hard as it looks now. So we'll worry about it later. Number 12. The graph shows the effect of an enzyme on a reaction. Okay? This is the time. This is the substrate energy. Of course, this is the graph for which type of energy?
Activation energy.
Yes. Which labels are correct? I want you to look at A, B, and C. Just give me 1 minute. I'll be back. Look at A, B, and C and decide what the answer is. Okay, I'm back. Now, I did not read the question. Who will give me the answer? Serene, you were there at, I think, when we talked about activation energy. Which label is correct?
Yes.
Excuse me?
This X is the reaction with the enzyme, right?
Let me see. X, this is X. They both have, X is with the enzyme. No, X is not the enzyme. The activation energy here was lowered. When it is lowered, it will be this graph. So this graph, which is Y, when I added the enzyme, X when there is no enzyme. The activation here is more. So there is more activation energy needed to start the reaction when there is no enzyme. When I added the enzyme, the activation energy was lowered. So this is the graph when there is an enzyme. Okay, Serene?
Okay.
Now, which label is correct? Let me see. X is the reaction without enzyme. Yes, okay, but W is the activation. No, W is wrong. A is right or wrong? Is W the activation energy?
No.
No, W is not the activation energy. Activation energy in W is all this distance. So A is wrong. Kenzie, tell me. B is the reaction with the enzyme. Sorry, X is the reaction with the enzyme. X is the reaction. Is this correct? Is X the reaction with the enzyme?
No.
Wrong. B is wrong. Very obvious. C, Abdullah. Y is the reaction with the enzyme. Y is the reaction. Okay. W is the difference in activation energy. Is C right or wrong?
It's right.
Yes, it's very obvious. Okay? W is the difference. In other words, all this distance minus this distance. So what W is the difference in activation energy. This is an easy question. Which statements about non-competitive enzyme inhibitors are correct? Okay, which statements about the non-competitive enzyme inhibitors are correct? The inhibitor can bind to the enzyme whether or not the enzyme has its substrate bound to in the active site. Is one right or wrong? Remember with me.
Right.
Right. The non-competitive inhibitor, it will bind to an alternative site. It does not bind with the active site. So it will bind to its alternative site whether the active site is bound to the substrate or not. Kenzie, tell me. The inhibitor may bind to an allostatic site. Right or wrong?
Is this the same of the alternative
Yes.
site?
Yes.
Yes, it's right.
Of course it's right. 3, the inhibitor may not prevent the substrate binding at the active site, but it will prevent product formation for sure. 3 is right or wrong?
Right.
It's also right. Maybe the substrate will bind, but will the reaction proceed?
No.
No, it will never proceed. So 1, 2, 3 has to be in the answer. Are you all with me?
Yes.
Okay. Did we do any mistakes?
No.
Good.
No.
Which of these statements describe the action of an extracellular enzyme? What is an extracellular enzyme? Remind me.
Secreted by the cell, or it acts outside the cell.
Yes, the enzyme is outside the cell. So let's see. 1, synthesis of a polypeptide in the nucleus during DNA replication. Is 1 right or wrong? This enzyme, can it be in the nucleus, Abdullah?
Yes, miss.
No, pay attention with me. This is so important. Answer number 14, the question in front of you.
14.
Yes, 14, yes. Which of these statements describe the action of an extracellular enzyme? An extracellular enzyme, Abdullah, works outside the cell or inside the cell?
Outside.
Very good. So can number 1 be correct? The synthesis of a polypeptide in the nucleus. In the nucleus, can it be in the nucleus?
No.
No. So 1 is wrong. So this will make me exclude A and B. I will not even look at B. Let's look at number 2. The digestion of macromolecules in the lumen of the small intestine. 2 is right or wrong? Yes, I'm outside a cell, so 2 is right. So this means 2 has to be in the answer. So I am still confused between C and D. 3, synthesis of ATP molecules in the mitochondria. This means inside the cell. Is 3 right or wrong?
Wrong.
Wrong. So the answer has to be only 2. Okay? Farouk, I can't. Huh?
Yes, I can't understand why 1 is right.
1 is wrong. The synthesis of a polypeptide in the nucleus. The nucleus is inside the cell, and this enzyme is extracellular. It cannot be inside the nucleus.
Okay.
Okay. Now, Farouk, I want to hear your voice, please. Answer this question.
Yes,
An experiment was carried out in which the enzyme lipase was used to hydrolyze a triglyceride. Triglyceride means a fat. Lipase will break down fats. The pH was recorded at regular intervals during the reaction. The results are as shown. So this is the time, and they recorded the pH at every 2 minutes, obviously. The pH obviously decreased, and then it stopped at 4.6. Now tell me, Farouk, when fat is broken down, it is broken down to give you what?
Fatty acids and glycerol.
Very good, fatty acid and glycerol. Fatty acid is an acid. So what do you think the fatty acid will do to the pH of the solution?
Decrease it.
Decrease it. So the pH is decreasing. When the pH is decreasing, how will this affect the enzyme itself?
Denature.
Yes, you are very good. Let's read the question. At 14 minutes, unreacted triglyceride was still present. So this means that the reaction was complete or not complete. What explains the results after 10 minutes?
Not complete.
It's not complete.
Okay.
Yes. Okay, choose. A, B, C, or D? What explains the results after 10 minutes?
D, because it's denatured.
Excellent. When you understand the question, you will easily get the answer. Does anyone has any objections? We said that fatty acid will be produced as the fat is being broken down. Low pH means that the enzyme will be denatured. So that is why at 14 minutes, unreacted triglyceride was still present because it was denatured. The moment we remember or when we write or mention the word denature, we have to mention that the shape has been changed. The tertiary structure of the enzyme is changed. No active site, it changed. Okay, so the answer is D. Any objections? Anyone who has any questions? Okay, who is not speaking today? Kenzie. Hello,
Yes, doctor.
Kenzie. Tell me number 16. Which statement about the effect of substrate concentration on the activity of an enzyme is correct? Remember the equation or the graph, sorry. Substrate concentration on the x-axis and the rate on the y-axis as the graph looks like this. Okay, so above a certain concentration of substrate, an enzyme reaches its maximum rate of reaction. A is right or wrong?
I think it's right.
Yes. Remember we said at this part of the graph, what was the limiting factor?
The substrate concentration.
Very good. And then here, what happens? Why is it now constant?
Because it's no more limiting factor.
Then what is the limiting factor here? If the
Enzyme
substrate,
concentration.
yes. So A is correct. Okay. If you want to make sure that B and C and D are wrong, you can read. Let's read. At high concentration of competitive inhibitor, increasing the substrate concentration has no effect. Is this right? It's wrong. We keep on saying that increasing the substrate concentration will cancel out the effect of the competitive inhibitor. So the B is wrong. C, at high substrate concentration, a non-competitive inhibitor no longer affects the enzyme.
It's wrong, but
Of course, any inhibitor affects the enzyme. D, the higher the concentration of substrate, the faster an enzyme can catalyze a reaction.
it's wrong.
It's true until a certain point. After that, more substrate concentration will not make any effect. So did we do any mistakes or not yet?
Not yet.
Not yet? Let's do a mistake. Okay, number 13. Number 13, an experiment was conducted to investigate the effect of temperature on the activity of this enzyme, beta-glucosidase. The enzyme was tested when the solution was free and when immobilized in alginate beads. The results as shown. So this is the activity of the enzyme here on the y-axis and the temperature. And obviously we will start with a high activity and then the activity will drop as the temperature increases. And look at the key. This is immobilized, and this is the free. Which statement about the effect of immobilization is correct? Someone read and tell me. You tell me. I will not choose. Very easy. Very easy. Immobilization, what does it do? Farouk, when the enzyme is immobilized, it cannot move. What are the advantages? One of the advantages are right there. A, B, C, or D? Farouk, where did you go?
Yes. I don't remember this else.
No, remember. When the enzyme is stable, when it is fixed, what are the advantages? Does it denature quickly?
No.
No, it is more stable, more temperature stable, more pH stable. It has more shelf life. I can reuse it. It's not contaminated. I won't need to filter the product. I told you to memorize this list. So the answer is definitely D. Anyone has any objections? This is an easy question. Mariam, this question is for you. An investigation was carried out into the effect of an increasing concentration of substrate molecules on the rate of an enzyme catalyzed reaction. All other variables were standardized. Which statement is correct? This is my graph. Substrate and reaction rate. My pen is not working. Mariam, tell me what's the answer? A, B, C, or D?
A.
Yeah, it's very easy. The rate increases to a maximum and then it levels off. It's easy if you remember the shape of the graph. If you study well and you remember the shape of the graph, A is definitely correct. I will not even need to read the rest. But if I want to be sure, B, C, and D must be incorrect. The rate increases to an optimum and then decreases. Does the graph look like this? Year 11, are you with me?
No.
No, so B is wrong. The value of K of Michaelis constant will increase. Does the value of Michaelis constant change? Did we even mention anything about inhibitors?
No.
No, so the Michaelis constant value will be fixed. It will not increase or decrease. The Vmax will never be reached. Is this correct?
No.
No. So we are now sure that A is right, B, C, and D, the three of them are wrong. Anyone has any questions? Anyone has any questions?
No.
Okay. Which features, Serene, let's answer this question. Which features are correct for a competitive inhibitor? You were not in class. You did not attend inhibitors, yes?
Yes.
Okay, Kenzie. Which features are correct for a competitive inhibitor of an enzyme catalyzed reaction? A, B, C, or D?
I think it's A.
Very good. It is A. Why did you choose A?
Because the inhibitor didn't change the shape of the enzyme.
Yes. Okay, but still, it can be A or B. Why is it not B?
Because it's not similar shape to substrate.
Yes, it is a similar shape. Very good. This is an easy question. Also, it's easy if you studied well and you remembered the competitive inhibitor. Okay. This question is for who? Who do I have here? Where is Yassine? Where is Abdullah? Okay, Abdullah, this question number 16 is a little bit tricky. I remember this question from last year. Liver cells contain membrane-bound organelles called peroxisomes, which contain the enzyme catalase. This enzyme hydrolyzes hydrogen peroxide to water and oxygen. A student cut two identical sized pieces of liver and placed one in the refrigerator at 5 degrees and one in a freezer at -18. Okay? After 12 hours, both pieces were raised to room temperature and placed in equal volumes of hydrogen peroxide. If you were not paying attention, what did they do? They got two pieces of liver and then they put one in the fridge and one in the freezer, and then they got them out and they both were heated to the same temperature, and then they put them in a beaker containing hydrogen peroxide. The liver that had been at -18 degrees produced bubbles of oxygen more rapidly than the liver that had been at 5 degrees. Which statement explains why the liver that has been in the freezer will produce more bubbles of oxygen than the liver that has been in the fridge? Can you guess why? Abdullah, are you with me?
Yes, I'm reading the answers.
Okay, read. More bubbles, Abdullah, means more enzyme or less enzyme was released from the liver.
More.
Of course, more bubbles means more enzyme was released from the liver to react with the hydrogen peroxide. And they told you that that piece of liver, when it was put in the freezer, it gave us more bubbles. So what did the freezing do to the liver?
Damage the cells.
Yes, you are right. Freezing will damage the cell membrane. The cell membrane will control what will move in and out. When the cell membrane is damaged, the enzyme will freely move out. When it goes out, it will bind with the hydrogen peroxide or react with it and produce more oxygen. So the answer is C. Okay? What are we correct? This is 16C. Did we do mistakes or not yet? I'm not correcting. Did we do mistakes? Someone tell me. Did we do mistakes or not yet?
Not yet.
Okay, we will continue until we do a mistake. Let's do one more exam and then I'll let you go. Where are we? Yeah, here. June 2018. Good. Which words from the table correctly complete the paragraph about enzymes? Kenzie, no one will be able to answer this question because we did not take the bonds yet. Let me answer it for you. When the pH of an environment is decreased below the optimum pH of an enzyme, the bonds between the adjacent whatever groups holding the structure are disrupted. But you can know number 3. What is the name of the structure of the enzyme?
Tertiary.
Yes,
Tertiary.
structure. It's definitely A. Okay, we will study the bonds, and these bonds are hydrogen and ionic and disulfide and what's this called? Hydrophobic bonds, and they happen between the R groups. You know what I explained at the beginning of the lesson? So 12 is A blindly, okay? Because of this term, this tertiary structure, because an enzyme is a tertiary protein. The effect of substrate concentration on an enzyme catalyzed reaction was measured in 3 different conditions: with no inhibitor, with a competitive inhibitor, and with a non-competitive inhibitor. This is the graph that we drew in class, exactly this graph. The graph shows the results. Which statement is correct? Kenzie, read and tell me which is correct. Inhibitor, tell me Kenzie, inhibitor X is competitive or non-competitive?
It's competitive.
Shatura. And inhibitor Y?
Non-competitive.
Excellent. Now, which statement is correct?
Yes. Wait a second.
I'm waiting.
It's D.
Yeah, it is D. Y is non-competitive inhibitor which binds to a site other than the active site. Again, a nice straightforward question. Okay, we did mistakes or not yet?
Not yet.
Okay, let's do a mistake so that we finish. A student investigated the hydrolysis of the lipid in high-fat milk using the enzyme lipase. Okay, 1 centimeter cubed of the enzyme solution was added to 10 centimeter cubed of high-fat milk. The temperature was constant. The pH of the reaction mixture was recorded at time 0 and every minute for 20 minutes. Which statements could be supported by the results of this investigation? I advise you to always remember that when lipid is involved in a reaction, lipids will be broken down to what? What are the final products of
Fatty acid and glycerin.
fatty acid? Fatty acid and glycerol. And fatty acid always lowers the pH. And when the pH is lowered too much and it moves away from the optimum, what happens to the enzyme?
Then slows down.
Yes, okay. So which statements are supported? Less product is made as time proceeds because the substrate is decreasing. Is one right or wrong?
Right?
Yes, one is right, of course. 2, the pH of the reaction mixture changes more rapidly in the first few minutes and then changes less rapidly. In other words, they're talking about the initial reaction rate.
Yes, it's right.
The initial reaction rate is always fastest at the beginning because at the beginning we have enough substrate. The substrate is not a limiting factor. So 2 is correct. Are you all with me or I'm moving too fast?
No.
Okay. I can hear only one voice. Anyone has any objections? Please stop me. 3, the product gradually causes more lipase molecules to denature. What do you think, Abdullah? The fatty acid and the glycerol will cause the enzyme to denature or not? Abdullah,
I was muted. Okay.
Abdullah, why do you daydream? Can you focus?
No, I was talking, but I forgot to unmute my microphone.
Okay, don't talk. The product, when we break down fats into fatty acid and glycerol, the fatty acid and glycerol will gradually cause more lipase molecules to denature or not?
Yes.
Yes, so 3 is right. So what's the answer?
A.
A. Okay. Mariam. A student carried out an investigation into the effect of temperature on the rate of an enzyme catalyzed reaction. At each temperature, the substrate concentration was measured after 10 minutes. All the other variables were kept constant. Which graph shows the effect of an increasing temperature on the substrate concentration after 10 minutes? Now, I want you to pay attention Temperature is on the x-axis, but the y-axis is not the reaction rate like we are used to. It is the substrate concentration, not the reaction rate. The graph that we are used to when we have the temperature on the y-axis and the rate of the reaction on the, when we have the temperature on the x-axis and the rate on the y-axis, the graph looks like this. But we don't have the rate on the y-axis. We have the concentration of substrate. So think about it. How will the graph look like, Mariam?
D.
Correct. Did you look at the mark scheme ولا انتي شطورة؟
No, I didn't look.
شطورة. The substrate concentration as the reaction is proceeding will decrease, 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 decrease. Okay? And then at very high temperature, the enzyme will be denatured. Will the substrate be used in the reaction after the enzyme is denatured?
No.
No. So that's why the substrate will increase again. Think that the substrate, put in mind, the substrate is produced by the body. The body is producing substrate, producing substrate. When the enzyme is there, the substrate will be broken down. So the concentration of the substrate will decrease, but the temperature is increasing. The enzyme will denature. So the substrate will increase and there will be no enzyme to break it down. That's why the graph is D. Do you all agree with Mariam?
Yes.
Okay, Farouk. The graph shows how the rate of an enzyme catalyzed reaction depends on the concentration of substrate. I have the substrate concentration on the y-axis and the initial rate of the reaction on the, I have the substrate concentration on the x, and the initial rate of the reaction on the y, just like the one we drew in class. And the graph is the typical graph. What is the Michaelis-Menten constant? Very easy question. Farouk, how will you get it? Farouk, where did he go? He's not here. Abdullah, how will you get the Michaelis constant?
Dividing the Vmax by 2.
Okay, the Vmax is 3.2, 3.4, 3.6, 3.8. 3.8 divided by 2. Use your calculator. What is 3.8 divided by 2? Let's do it together. And after,
1.9.
excuse me?
Doctor, it's 0.38, not 3.8.
0.38. شطورة. صح. 0.38 divided by 2. What's the answer?
0.19.
How much?
0.19.
Okay, then it is 0.19. Will be somewhere here, yeah? Then after we go here, Abdullah, what do we do?
We measure the substrate concentration at the point of the half Vmax.
Yes, and what do we get?
1.5.
Yes, is the answer C? 14, is it C? Yes. The first two, did we get them correctly?
Yes.
Okay,
Yes.
enough, or you want to solve another exam? Let's vote. Abdullah, you want another exam? Or enough?
Enough.
Okay, Mariam, enough or another exam?
Enough.
Oh, okay. Serene,
Enough.
okay, Kenzie,
Enough.
okay. You will do the rest on your own?
Yes.
Promise?
Yes, and it will be submitted.
Oh, okay, we will see. Fine. Paper 1 is not very nice. I think you realize this by now, so it needs our attention, okay? Paper 2 is easier, but paper 2, we solve it after we study very, very well. We look at the notes, we get the answer from the notes because the mark scheme is not the answer. The mark scheme is just an outline. The mark scheme tells you what should be in the answer, okay? Now I'll let you go. Have a good day and see you next Monday. Bye-bye.
Yes, miss.
Yes, my dear.
Miss, هو if I studied from the notes يعني يعني هعرف أفهم يعني will it be enough without يعني any explanation?
Well, I try my best to write notes that are easy to understand. So this is your decision. Some students, they study from biology books and they don't need any explanation. Some students, they need someone to explain to them. So it depends on the student himself. You are home because you have an injury and you cannot come, yes?
Yes.
So of course you have to not waste any time and try to study from the notes. And if you have any question, ask me.
Okay.
Of course, explanation helps much, like explanation makes life very easy. But now you have a situation. When can you come back to school?
After a month.
Oh my God.
Mm.
I'm very sorry. I hope you a really, really quick recovery. I advise you to take vitamin C because it makes healing very fast.
Okay.
What happened?
I was at my training في التمرين ورجلي اتعوجت فجسمي حمل عليها ورجلي حصل لها شرخ.
Oh, basketball?
No. Football.
Oh, really? Okay. You like football. I usually see boys liking more football. طب ألف سلامة عليكي. والله I'm very, very upset. But like, don't waste your time. Read as much.
أيوة ما I'm trying عشان كده I attended today's class.
Yes, of course. And this class is going to stay fixed every Thursday to solve paper one because paper one is giving us a hard time. Okay, my dear, if you need anything, I'm always here to help you.
ماشي. Okay.
Bye bye.